Yes, sir. It is very clear, sir.
Yeah.
Yes, thank you very much, sir. Thank you very much. We will start then, sir. We will start with.
Yeah.
Yeah, thank you very much, sir. 1 minute.
Sure. Sure.
Yes,
Ma'am.
yes, 1 minute, sir. Sir, kindly unshare now, sir. So please wait for 2 minutes, sir. Parvati, is it streaming? I wanted to know. Yesterday's. Now live session has started. That as it started streaming. Enter through the link. Enter through the link. Good afternoon, participants. I welcome all of you to the Day 2 session of our international webinar series. Now I kindly invite Dr. Savita Murthy, Assistant Professor, Department of Botany, Mount Carmel College, to take over as the moderator of today's sessions. Savita ma'am, over to you.
Yes, thank you, Jayanti. Good afternoon, all. I welcome all the participants, speakers, and my dear colleagues for the second day of International Webinar Series on Secret Treasures in Plant Science. Now, I request Dr. Roshni Rao to introduce the first speaker. Over to Roshni ma'am. Roshni ma'am.
Hello, can you hear me?
Yes, ma'am.
Yes, good afternoon, one and all present there. It is my privilege to introduce to you Dr. Pasupuleti Vishweshwar Rao. Dr. Rao is currently working as a faculty in the Medicine and Health Sciences University, Sabah, Malaysia. He is head of non-communicable diseases research area and also the faculty research coordinator. Prior to this, he has held several important posts. He was the director of Institute of Food and Security and Sustainable Agriculture, chairman of Animal Ethics Committee at University of Malaysia, Kelantan, a visiting researcher to Japan, Macau, Indonesia and Thailand. He has also worked at several different universities in India and South Korea. Dr. Vishweshwar Rao has 14 years of teaching and research experience in the fields of biotechnology, nanoparticles, environmental engineering, food and agriculture, all of which have a very important and applied aspect. Dr. P.V. Rao has been elected as an Executive Council member and co-chairman of International Networking Group at Young Scientists Network in the Academy of Sciences, Malaysia for the year 2018 and 19. He has published more than 70 papers in international journals, written 6 book chapters with reputed book publishers like Springer and Elsevier. He has also been an editorial member and reviewer for several high impact factor journals, including Nature Publishing Group. Dr. Vishweshwar Rao has been awarded a certificate from the Elsevier for his outstanding reviewer contribution, and he has also conducted several workshops in Malaysia and abroad. His topic today is very interesting. It is phyto nanoparticles and its application in biomedical sciences. We are very lucky to have such a reputed and meritorious scientist to deliver a talk in our webinar today. Welcome to you, sir, and over to you.
For your kind words and nice introduction. So are the slides visible?
Sir, now
No,
you
sir.
can share your screen, sir.
Yeah, please do let me know once the slides are visible.
Sir, slide is visible, sir. Kindly put it on the slideshow mode, sir. Yes, sir. It is fine now, sir.
Yeah,
Yeah, thank
but
you.
I'm not sure. I don't know. When I press the video,
Yes, sir.
it is not accessible. If the organizer
No, sir, but it is clear now. Your
is
video.
clear, right?
Yes, yes. You can go ahead, sir. Thank you.
Okay, thank you so much, Dr. Roshni ma'am and Dr. Jayanti and all the organizers for having me here with this international webinar series on secret treasures in plant sciences. And my topic is all about nanoparticle research, since Dr. Roshni ma'am has clearly mentioned about my areas of expertise. And usually I'm working on nanoparticles and also other biomedical applications. So let us see what exactly is nano. So nano is, for me, nano is not a new thing. Nano is an old phenomenon which is existing in nature and which is also been in practice since ages, but now, based on the wide variety of applications, we are focusing into this particular field in a wide variety of areas, including agriculture, food sciences, medicine, engineering, whatnot. So if you can observe nowadays, including the cosmetics, if you observe the cosmetics, they also have nanoparticle sciences and also nanoparticle technology, which have been used very tremendously in the nature. And what is nano exactly? What kind of particles we call as nano? Is it very huge or it's very small? Nano is actually, you can see this picture, nano is very, very, very small particles which we cannot see with our naked eyes. If you can say 1/1000th of meter, or in a layman language, we can say that if you cut a hair, human hair into 1,000 times, then you can get a nanoparticle which is in the range of 1 to 100 nanometer in range. Then we can consider it as a nanoparticle, which again I mentioned, we cannot see the nanoparticles with the naked eye. And there are various types of nanomaterials, nanoparticles with the source materials or source metals used, then the nanomaterials will be changed accordingly. Okay, this slide shows what exactly, as I was mentioning just now, whether the nanotechnology is a new one or not. It's basically a fundamental principle is that it's a combination of sciences, engineering, technology. Finally, it becomes nanotechnology or nanoparticle research. Nanoparticle research is nothing but we do the research using nanoparticles from various sources. So this is what I was explaining just now. It's all about 1 to 100 nanometers in range and at least one dimension, and involve developing materials or devices within that particular size. And when you take the material or nanoparticles or nanomaterials which have been synthesized using nanoparticles, will be with very good stability and very good strength. That is why the researchers and also the public always try to go for nanotechnology related materials which long last. The shelf life will be more in nanotechnology materials. See, in 1959, Richard Feynman, even in his talk, he has mentioned that he has not exactly used the nano, but he has brought up the concepts of nanotechnology, which means exactly what I have mentioned just now is not a new concept. But however, we, the researchers or academics or academicians or general public, we are looking the old technology in a new system. That is where, in my own words, I call it as a old picture in a new gallery. Usually people say old wine in a new bottle, but here I wish to say that old picture in a new gallery, which means we are still talking about the old things, but the applications, the advances in this particular field is really vast and it's really amazing. See, in 1974, a Japanese professor, Norio Taniguchi, also mentioned it consists of various steps: processing of processing, separation, consolidation, and finally deformation. Whenever a nanomaterial you take, a nanoparticle, it forms in two different methods. One is top-down approach, another one is bottom-up approach, which means any molecule, any atom you take, it forms, building up. There are lots of building blocks to form an atom. So when an atom is formed, there are so many molecules, so many small ionic molecules, linked together and finally form a particular atom. And processing of separation, consolidation, deformation, how exactly they happen, how exactly they have been involved in a particular nanoparticle or in synthesizing the nanomaterials or formulation of nanomaterials. Yeah, coming to the main core concept of my topic, because I don't want to really go into all type of nanotechnologies and all types of synthesis. So here this slide clearly shows you what exactly the methods available to synthesize the nanoparticles. Apart from this, there are so many methods even in physical and chemical, but I have clearly given you a few for your understanding: evaporation, condensation, and laser ablation in physical methods, and chemical methods. Here I have given Tech-Witch method and also BSS. Finally, if you observe at your right side of the slide, there are different types of biological methods, which is into my area, and my main focus is always into biological synthesis of nanoparticles, and we also call them as biogenic synthesis, because this is green approach, green technology and green methods. So here you observe the last part of the slide: microbial systems, fungal systems, algal systems, and plant systems. If further narrow down, this is actually the broad: microbial systems, fungal systems, algal systems, plant systems, all together comes under biological synthesis of nanoparticles. We use all these different types of sources, we call them as source materials, to synthesize nanoparticles using different metals. It can be silver, it can be gold, it can be titanium, it can be zinc oxide, whatever it may be. But in my case, I always go with the plant systems. Because why plant systems, you may ask? Why plant systems? Where there are different types of biological systems are existing. But I usually work with plant molecules or plant materials or plant extractions. Not only that, when you work with the plant materials, it is completely green. And second point, most important point, is cost effectiveness. And third one is availability. And fourth one is you can easily do the processing. So there are so many benefits using plant extracts or plant materials or plant parts as the source material to synthesize the nanoparticles. And here you can observe, as I was mentioning, when you synthesize the nanoparticles or any material for that matter, there are two different approaches. Number one is bottom-up approach, which means where smaller particles build up together and then forming a larger particle or larger molecule or larger atom, which means they form as a cluster, but still if they are within the range of 1 to 100 nanometers, we call them as nanoparticle. And whereas if you come to the top-down method, it is like cutting down the atoms from a big larger molecule. In scientifically we can say cutting down the macromolecule into micromolecule into nanoparticle or nanomolecule, which means again the particle or the molecule should stick to that particular range. So this is how exactly it forms when you use the metal ions, say for example silver nitrate or silver ions or gold ions or whatever the metals I have mentioned just now. You can take them as ions, and then you have to have the reducing agents, which means in plant materials we have, our plant materials consist various types of polyphenols, flavonoids, or combinedly we can mention them as secondary metabolites, which has the capacity to reduce the ions and to make them into the particles. If you can see in this particular picture, metal ions combined with these bio-reducing agents. Here in my case of scenario or in my research, I usually use plant materials as the bio-reducing agents or reducing agents. Since this is from the biological source, we call them as bio-reducing agents. And finally, it forms reduced silver atoms, which means here I'm talking about silver nanoparticles or gold nanoparticles, which means the metal ionic, the structure or the status of metal ions becomes the atoms, where it's been reduced using the plant materials as bio-reducing agents. And finally, you can observe this is how exactly it looks like. The plant materials form as a capping agent, and your ions will be tapped into the polyphenolics or bio-reducing agents. We call this as a capping, reduction, capping, growth, and stabilization. So why I want to mention here about stabilization? Because stabilization is one of the major important aspects, one of the most important aspects, I would say, in nanoparticle or nanomaterial synthesis. Once you get the nanomaterial, because the most important thing or the vital part of its synthesis is stability. If there is no stability in nanoparticles or nanomaterials, certainly the public or the market will not going to take that particular material, because whenever you purchase a thing, say for example, I have two different products, A and B. If you say I have A and B, A is normal and B is nano, but at the same time you should be able to highlight the benefits than the normal, what exactly you have in nanoparticle or nanomaterials or nano product. And as I have mentioned just now, medicinal plants. So my whole career since 14-15 years, I'm working in medicinal plants or herbal medicine. People say it as herbal medicine since I work in metabolic disorders, diabetes, cancers, and various types of metabolic disorders, etc. So this is what I have mentioned just now. The main reason to use medicinal plants due to their less or no side effects. Because if we observe, the synthetic drugs may have, or certainly they will have very simple to serious side effects, undesired side effects, we would say. And these are the fields nanotechnology advancements are really involving into. You can see food and agriculture, energy and environment, biotechnology, herbal medicine, textiles, medicine and healthcare, aerospace, robotics, information technology, and whatnot. Every field, each and every field, the nanotechnology has been occupied. Even cosmetics, nutraceuticals, pharmaceuticals, whatever the field you name it, you have nanotechnology there. So nanotechnology and nano advancements are really going into the fields. And if you take medicine, you talk about nanorobots. Nanorobots, they do most of the things, once we send into the human body. And another thing, now we talk about drug delivery, very targeted drug delivery systems. So nanotechnology is also useful in those particular kind of advancements. And these are, very simple things I have mentioned here. They are fundamental building blocks of nanotechnology and starting point for preparing nanostructure materials and devices. Their synthesis is an important research component, because when we talk about nanotechnology, when we explain about the applications and advancements and the benefits when compared to the normal routine methods, certainly people might appreciate, or even the researchers are more, attracting to this kind of research. So I have already mentioned about what kind of nanoparticles we can synthesize using plants as source materials. I have only mentioned here 4 different types of nanoparticles, but there are numerous types of nanoparticles or metallic ions can be converted into nanoparticles. So I usually and most of the times work with silver nanoparticles, sometimes gold nanoparticles, and zinc oxide nanoparticles. So since we use the metallic ions, we call them as metallic nanoparticles. And for your information, these are single metallic nanoparticles. There are bimetallic, which means 2 metal ions they use and then they synthesize the nanoparticles. They also have multiple metallic nanoparticles. When linking into, when you observe my topic, it's all about biomedical applications. Plant materials, I have clearly mentioned about plant materials, nanoparticle synthesis. Now we go to the applications. Before going into the applications, what kind of problems we have, or what kind of problems humans face, and what is the main culprit? For me, whatever the disease, this oxidative stress is the main culprit. Whatever the disease you take, or whatever the metabolic disorder you take, or whatever the communicable disease you take, whatever the non-communicable disease you take, oxidative stress plays a very bigger role than what we think of oxidative stress. Oxidative stress in a layman language is an imbalance situation within the cell. See, when in a very layman language, we can say, I am so stressed today, which means you don't have proper sleep, or you are so tired with lots of bundles of work, or there may be other external factors, or there may be other internal factors, which means stress is for the body and mind. Oxidative stress is for the cell. Cell is undergoing an imbalance or fluctuations in their redox reactions. That is what exactly we can say. If you observe here, it reflects an imbalance between the systemic manifestation of ROS, which is reactive oxygen species, or even reactive nitrogen species. These are all free radicals. And a biological system's ability to readily detoxify the reactive intermediates or to repair the resulting damage. That's what exactly the oxidative stress or the mechanisms of ROS and RNS, which means the toxic materials or free radicals, we call them as. Once they enter into our body, we have some natural antioxidants which may or which might detoxify them most of the times. If you do not have, or if an individual do not consist proper antioxidants within the biological system to fight with the oxidative stress or the toxic materials enter into the body, then the oxidative stress increases, which means the ROS or RNS increases it in the cell, and antioxidant status will go down. Usually antioxidants and ROS, RNS will be like this in a healthy cell. If you observe in a damaged cell, ROS will be higher and the antioxidants will be at lower level. So that's where we call it as imbalance in the cell. Anyways, we have that particular image here. So you can observe here antioxidants and pro-oxidants. If you have more antioxidants, then certainly you have good, your cells will be very active and you have very good system. If you have less antioxidants, especially during aging or when an individual has any disease or the metabolism is not functioning well, such kind of situations, the antioxidant status in the biological system would be certainly low. And when you don't have proper antioxidants in your body, the ROS and RNS or free radicals will try to encroach your cells and try to damage as much as they can. So this is how the imbalances will be. And in next picture, you can see different types of pictures. How exactly? This is what I have explained just now. If you have antioxidants and reactive oxygen species or free radicals in a equal quantity or proportion, so the cell can fight against the free radicals very promptly. And if you don't have proper antioxidants and you have excess of oxidative stress, there comes the headache. Certainly, the cells which consists of less antioxidants cannot fight as much as they have plenty of antioxidants. I hope you got it. And if further depleted antioxidants, if your system cannot fight with the free radicals, and moreover you are not providing any synthetic antioxidants, which means usually we take vitamin C's, and this is the right time we talk about vitamin C's and other antioxidants. Especially during COVID, people are asking to take more vitamins, more vitamin C especially, which is considered as natural antioxidant. So this is a very simple example of that. And here are some examples I would like to give for biological activities, which is from one of my papers. We have started with antimicrobial activities. We have synthesized here lots of things I am not focusing. After synthesis, you have lots of characterization. You have to characterize it physically. Even with naked eyes, you can just confirm whether the nanoparticles have been formed or not. And after that, you have to go for lots of characterization, spectral, electron microscopy, all those things. Here, I'm only focusing on the biological activities. Antimicrobial activities, once we prepare the nanoparticles, then we have checked with various types of microorganisms, especially, few of the bacterial strains and few of the fungal strains. And it has shown potentially very good activity. And later on, I thought to hypothesize, this picture is only hypothetically made, which and how the nanoparticles enters into the cell and how the cell lysis can be done. I have given 3 hypothetical mechanisms where even if you submit your manuscripts to very good peer-reviewed journals, they ask for mechanisms, and sometimes most of the times they ask for in vivo studies. And hopefully, if the participants were there in the yesterday's lecture, so they have clearly covered how exactly the big journals, treat the papers and they review the things. So that's why I have proposed these 3 different mechanisms, how exactly the nanoparticles interact with the enzymes, and finally how they try to inhibit the signal transduction, and it leads to the cell lysis, number 1. Number 2, it interacts in gram-negative bacteria, dephosphorylation of peptides, of tyrosine residues, cell lysis. And in the third mechanism, we have clearly depicted that interaction of the silver nanoparticles with cell wall, release of free radicals. Because just now we have mentioned about free radicals, how exactly they damage the cell. So the nanoparticles releases the free radical onto the cell, then they kill the bacterial cells. This is another mechanisms, various mechanisms. This is another picture with various mechanisms you can observe for apoptosis, how exactly it happens. So you can see in mitochondria, it produces ROS, reactive oxygen species, and oxidation of proteins, lipids, and DNA finally leads to apoptosis. So apoptosis will be the central principle where we target with the silver nanoparticles. Our main aim is to, do the cell death, or whatever objective we meant in the beginning, then we have to certainly achieve with our nanoparticles. This is what, and this I have taken from one of the papers, and uptake by endocytic vesicles. You can see in nucleus various types of mechanisms. Even you can see some of the caspases have been involved here. And finally, cell cycle inhibition. Once your cell cycle has been inhibited, finally anti-proliferation, and it leads to apoptosis as well. And finally, if you take the third mechanism, it's all about cytoskeletal injury and uptake by macrophages, activation of macrophages. Certainly, activation of macrophages increases the reactive oxygen species and finally leads to inflammation. And the fourth one, so it's laser assisted hypothermia, and fifth one is all about cell starvation, etc. And this one, this particular picture depicts about what kind of materials, or what kind of materials can be formed using nanotechnology. This is from one of my papers, which I've published in Central Drug Metabolism in 2014, where the nanotechnology and diabetes, or various types of nanotechnology and nanoproducts for diabetes was discussed. And we have clearly mentioned nanotubes, artificial pancreas, nanopumps, microspheres, nanoshells, quantum dots, layer by layer methods, fluorines, and smart tattoos. So these are all the things from various literature we have jotted into the into this particular picture. And further moving, smart tattoos. Here you can observe one of the aspects is smart tattoos. Another picture, this is the article which I was mentioning about with Current Drug Metabolism, one of the good journals from, If you observe smart tattoos from the previous picture, if you continue here, this is how exactly the smart tattoos look like. Okay, not necessarily look like this. So there are various types. Even nowadays, you observe various types of, or most of the people, they are wearing smart watches, which is with simple and similar kind of mechanism what we have hypothesized in this particular picture. It was in 2014, almost 6 years back. So the sensors are detected using a less invasive technology, and sensors are inserted under the skin. They usually tells, they keep monitoring your blood glucose levels. Once it is, the fluctuations in the blood glucose levels, the optical reader might, give you the information. And this is another one, the nanotechnology, various types of approaches for targeted delivery of therapeutics and also control of stem cell behavior. So since I wanted to give another example for neurological disorders, I have taken this one. Exactly, this is not from my works. Okay, the previous one was from my research. So hopefully I still have got time. And I would like to show some of the research focus in my lab. So this is what exactly I do. So my research started with natural product research, which means basically plants. And then I started working in metabolic disorders, especially diabetes. And then oxidative stress is always my core research topic. Whatever the disease I take, even diabetes, cardiovascular diseases, cancers, or whatever, oxidative stress would be the core component in my research. Antioxidants as well, because most of the time the secondary metabolites or polyphenols or flavonoids we take or we get from the plants, we use them as antioxidants. Then cardiovascular diseases, nanoparticle research. Whatever I do, whatever the research I plan, I execute everything towards human wellness. At the end of the day, it should link or it should give at least a basic answer. If you do some fundamental work, some basic answer to the research questions. So far, I have used various types of plant materials and even honey to synthesize the nanoparticles and also for the metabolic disorders. So this is one of the papers we have published with stingless bee honey. Even since past decade, I am working on honeys, various types of honeys. Even in Malaysia, stingless bee honey is one of the popular and prominent honeys. And most of the time nowadays, I work on stingless bee honeys, various types. So I have executed some of the research with 5-6 different types of stingless bee honeys. And this is Rhinacanthin C, which we have isolated, and finally we have executed the anti-diabetic activities. Relating to this topic, this is one of the very premier plant material or plant from Malaysia. They call it as Malaysian ginseng, Eurocoma longifolia. So in a local terminology, they call it as Tongka Ali. So which is used for various types of health benefits. Even the layman, trusted this particular plant for various types of biological activities. But I have synthesized the nanoparticles, silver nanoparticles, and then we have tested for antioxidant activity. Even the antioxidant activity was also not reduced after forming the silver nanoparticles. Usually, if the nanoparticles consist good stability, it will certainly have the antioxidant activities intact. And medicinal plants, this is a simple review I wanted to, shed the light on. And I have clearly focused on various types of biological benefits. As I have mentioned earlier, we even work with different fields, engineering, agriculture. Since my previous organization, I was completely working on various aspects of food security, agriculture, but my main area is into biochemistry. So here we worked on various applications or various crops, especially the silver nanoparticles, on growth and biochemical parameters of rice, maize, and peanuts. The prominent results have been seen. And this is one of the reviews, phytochemicals and biogenic metallic nanoparticles as anti-cancer agent. This article mainly discusses about various types of mechanisms, antioxidant, anti-cancer mechanisms, how exactly they enter into the cells and other stuff. So this is how usually the nanoparticles been made, plant materials and functional groups. Even to confirm the functional groups, we use FTIR to characterize, and when it is combined with silver ions, finally it forms silver nanoparticles. Then the silver nanoparticles we can use for various types of activities, biological activities. And this is how it looks. So this slide should be in the very beginning. So the plant materials, metal salts, and it forms nanoparticles. These are all characterizations: UV, XRD, FTIR, ACM or TAM, particle size analyzer. So these are all various types of characterizations we do. And finally, we go for various types of biomedical applications. If I design one research for antibacterial assay, then certainly we go for antibacterial. Not exactly everything. One plant extract cannot go for all the activities. So it depends on the phytochemicals available in the plant materials. So we know nanotechnology will change the world. It's time to think about how. So it's by Alan Leo, MIT. Get ready for your nano future. In conclusion, not only the plant materials, even the nanoparticles synthesized biogenically or using green technology, the nanoparticles play a big role or vital role in the human health, which ameliorates various types of diseases, not only diseases and their complications. Say, for example, you take diabetes, they have lots of complications like neuropathy, nephropathy, retinopathy, cardiomyopathy, etc. So they also try to ameliorate through various pathways. Here we have not discussed much about pathways, biochemical pathways. So they try to, ameliorate the things in various types of pathways and then try to reduce the damage to the cells, and hopefully based on the capacity of the plant materials, based on the capacity of the nanoparticles, the recovery depends on. So these are my research team. So a few more are, missing here. And these are my collaborators. And this is one of my most memorable moment in my life, apart from lots of awards. So this is one of the Grand Alumni Award I have received from one of the universities from Bangalore, which is Reba University. So thank you for giving me this opportunity, and thanks for having me here.
Yeah, thank you very much, sir, for your excellent talk. Now we'll take a few questions from the participants. The first question is from Arpita, requesting sir to kindly suggest suitable journals for publishing work on genotoxicity of gold nanoparticles in plants and suitable institutions for postdoc research on nanotech in plants. Sir?
Okay, that's a very good question. So it's related to the journal. So maybe you can target some of the journals. Say, for example, International Journal of Nanomedicine, which is from Dow Press. But if you have if you have support for article processing charges, you can go for that. Otherwise, there are lots of nano journals, named journals we call them as. You can submit to Nano Science or Nanomedicine, which is from Elsevier, and Nano Research. You can also submit to Nano Research, or you can have a look into Taylor and Francis, one of the journals named as Artificial Cells Nanobiotechnology. So you can submit to that. Even the impact factor is also very good. That is a very standard journal. Your first question. Second question, what are the institutions for postdoc? If you are looking into Malaysia, probably you can link, you can have a look into different government organizations which have good nano labs. And if you ask me, in Singapore, you can look into A-Star, NUS, NTU. And US and Europe, it's up to you, because various countries, they have various types of advancements. Even Australia, they have very good nanotechnology advancements. So you can try these things. I mean, I have given you some examples in Asia, Europe, and the US.
Okay, thank you. The next question is from Dr. Guriraj Hbe. Most of the nanotechnological research are not commercialized. When used in therapeutics, is there any example of a nano delivery system which is successfully adopted for treating various cancers?
Yeah, that's a very, very enlightening question, I would say, because that's the major debate going on across the globe about nano delivery and the successful rate. Because as of now, very few researchers have gone up to the commercial level and also into the doable level. That is what I can say. So which means which are in practice very minimal technologies or very minimal nano research has been practiced at a very minimal level. Hopefully there is a big future. Even now we had a discussion very recently with one of the countries related to COVID, and they were also mentioning that we want to look into nano drugs. So hopefully there might be lots of technologies which will be available for humans with a less cost if more focus towards nanotechnology.
Okay, thank you, sir. Next question. Sir, is there any specific method to synthesize silver nitrate or zinc oxide nanoparticle from plant materials, or any specific instrument to carry out this research in laboratory? This question is from Dr. Suraj.
Yeah, thanks, Dr. Suraj. You have asked two questions here. One is about which kind of method you can use and what type of characterization you can do. So number one, you can use different methods. Say, for example, some of the researchers, they use boiling methods, which means they just boil the plant extracts and then they do some formulations and then mix with the silver metals or say silver nitrate. Finally, they try to establish the synthesis. Or some people, they don't go with the boiling methods. They approach differently. And some other researchers, they use another method with using direct plant materials. So there are various methods which you can approach according to your feasibility and flexibility, I would say. And coming to your next question is about characterization. So once you synthesize the nanoparticles, the first and foremost thing we have to confirm whether the nanoparticles have formed or not, which means with your naked eye you can confirm physically whether the nanoparticles have been formed or not. But to scientifically establish or scientifically say that whether the nanoparticles have been formed or not, you have to go with UVVs, spectrophotometer or photometry. Then you have to go for various characterizations as I shown in my slides. One of the slides is XRD, FTIR for functional groups, XRD for nature, crystalline nature, and SEM or TEM to know the molecular, the size, particle size, or even you can go to zeta potential and particle size analyzer with the facilities. Otherwise, collaboration is only the way. I mean, here I'm not sure the participants, level of participants, even early career researchers or even research scholars, I would say collaboration or collaborating with the people whom you know might give you better fruits, than working alone. So if we don't have facilities, and certainly there is a way to collaborate with the people and do the great jobs. Yeah.
Okay, sir. Next question from Rashmi Pallavi. How could plant extracts act as anti-agglomeration for synthesis of nanoparticles?
Okay. How do plant extracts act as anti-agglomerative agents?
Yeah, that is what she asked, I think.
Yeah. So agglomeration usually depends on how we do the experiment. See, sometimes we observe the agglomeration very much. I mean, based on the disturbances in the peaks, you can observe there is an agglomeration. Even when you observe your nanoparticles, you can observe the agglomeration. When you go for SEM, there is an agglomeration. But agglomeration is most of the times how we do, number one. Number two is all about dispersion and distribution. It all comes with the synthesis. If the synthesis is being done properly, then the dispersion and distribution of the, if you remember from my slides, the bioreduction. If the bioreduction occur very clearly, then there might be less possibility for the agglomeration. So if the process has not been followed thoroughly, then certainly there will be possibility for the agglomeration. So it's all about how we do the experiment and how care we are taking not to have these kind of errors in the experiment. So hopefully you can have with that mainly dispersion and distribution, and the plant materials or plant molecules or phyto chemicals, they have the capacity to bioreduce the silver ions or metallic ions, and then they cap the materials and then finally form the nanoparticles. So agglomeration is all about distribution and dispersion.
Okay, sir. This is from many participants. Can the nanoparticles kill viruses?
Well, that's a good question. Yeah, if you observe some of the literature, the viruses also can be, inhibited by nanoparticles. Some of the research, because I have never worked with viruses, so I worked with bacteria, fungi, and other sorts of, non-communicable, what we can say, microorganisms. So viruses, yes, of course, there are various reports saying that nanoparticles has the capacity to kill the viruses.
Okay,
but the mechanisms, certainly we have to explore into the detailed literature.
Okay, sir. Question is, how nanotechnology can be used to reduce the impact of climate change in agriculture?
Wow, that's a very good question. Even very similar question been asked very recently. Well, this is again a question very much related to the agriculture and climatic change. Again, this question is related to the toxicity. This particular topic I have not covered here, but anyways, usually there is a very big debate going on the nanoparticles and toxicity towards climate and also to the agriculture crops. But when we use different types of concentrations which are really biodegradable and which can be easily, benefit the crops. Say, for example, even we use various types of nanoparticles onto the crops for the development of growth. If you observe one of my slides, we have used nanoparticles, different formulations and different concentrations for the growth, to check the growth and to check the yield for 3 different crops. We have used in rice, maize, and another crop. So, where we have used very less concentrations in order to save the climate. in order to not to have climatic changes or any disruption to the climatic changes or to the crop fields. So which means always it depends on the concentrations we use and always the type of molecules you have in your nanoparticles. And on top of that, we always have to check the toxicity of the nanoparticles before we apply into the nature. So that's how we avoid, damaging the climate or environment or polluting the environment.
Okay,
so
sir.
hope this answer helps you.
Yeah. Sir, another question. Is there any scope for MSc Botany students in field of plant nanotechnology?
Yes, of course. So plant biotechnology, see, whatever we take, we evolve, even we talk microbiology, biotechnology, or other advanced subjects, nanotechnology, whatever, everything evolved from a basic botany, zoology, chemistry, physics. These are the father and mothers for whatever the technologies we are using nowadays. So which means if you have the basic degree in botany or zoology or chemistry or physics, any specific field can enter into nanotechnology. That's where I have mentioned very clearly, nanotechnology is everywhere. So and certainly botany students can enter into phyto nanotechnology or nanotechnology related to the plant sciences.
Okay, this is the last question. What is the aspect ratio of nanoparticle and how can I calculate it? Dr. Suresh Babu has asked this question.
Okay, that's good. Can you repeat the question, madam?
Yes, sir. What is the aspect ratio of nanoparticle? And
Sorry, what is the? No, the voice is not clear.
Aspect ratio.
Okay, aspect ratio
of nanoparticle and how can I calculate it?
Okay. So thanks, Dr. Suresh Babu, for asking the technical question. Very good. So there are different types of calculations you can calculate the aspect ratios, and aspect ratios usually depends on your particular materials and particular source materials, how exactly you take them. So based on that, you can try to calculate your aspect ratios, or if you need more information, you can just write to me. Even you can take my email ID from the organizers, so I can give you more information on that, or even I can send you the related documents to you.
Okay, thank you. Thank you very much, sir, for your excellent presentation. Due to time constraint, there are many more questions, but due to time constraint, we are not able to ask all the questions. Thank you very much, sir.
Yeah, thank you so much, all the organizers and also Mount Carmel College for having me here for this international webinar series on secret treasures in plant sciences. So any of the participants or even the organizers would like to collaborate, I'm very much, you're very much welcome. So you can just Google my name. So there are many papers. So you can just write to me. My email ID will be there. So that's the easiest way we can communicate with each other. If you need any information about my research or my, any information. Yeah. Thank you.
Thank you very much.
Yeah. So I shall.
Sir, please. Rao, sir.
So I will be leaving the session now, since I have to attend another meeting.
Okay, sir. Yeah, thank you very much, sir. Thank you.
Thank you so much. Yeah.
Dear participants, now we'll move to the second session. It's my privilege to introduce today's second session speaker, Dr. Ramesh Kumar Saini. Sir has finished his bachelor's and master's degree in agriculture from GB Pant University of Agriculture and Technology, Pantnagar, India. He received his doctoral degree in biotechnology from CSIR institution CFTRI, University of Mysore, India. Currently, he is working in the Department of Crop Science of Konkuk University, Korea, as Associate Professor. He is recipient of MENT scholarship from Department of Biotechnology, Government of India, and also he has completed his PDF from Department of Biotechnology, Government of India. To his credentials, he has published more than 40 research papers in peer-reviewed national and international journals with the total impact factor of 169.744. Currently, he is working on a project entitled Value Addition of Shrimp Processing Waste via Efficient Recovery of Commercially Vital Carotenoid Astaxanthin and its micro encapsulation for enhanced physical and biological activities, funded by National Research Foundation of Korea. Sir's primary research interests include development and validation of analytical methods for the identification and quantification of nutraceuticals in food, cytotoxic and anti-proliferative potential of natural carotenoids against various cancer cells. With this brief introduction, I request Dr. Ramesh to deliver the lecture. Over to you, sir.
Can you hear me, please?
Yes, sir, we can hear you, sir.
Thank you, Savita ma'am, for your overwhelming introduction. Okay, can you hear me?
Yes, sir, we can hear you, sir.
Okay, I can go now?
Yes, sir, go ahead, sir.
Thank you so much for considering me for this very important webinar, Hidden Treasures in Plant Science. And today, I'd like to talk on the industrial potential of carotenoid pigments from plants, current trends, and the prospects. So first, I want to tell you that why I am here and why I am presenting on this topic. So when you search on the Web of Science, any paper published on the word carotenoid in the title in the last 3 years, when you search in the Web of Science, 1,430 articles will appear. And when you sort this article on the basis of the impact factor or the usage count, the rank 1 article is published by us from our lab. So we have published so many articles in a great journals which received so many citations, and you may find many articles in this list. So with the experience of the 12 years experience on this field and the carotenoid, we came across so many interesting facts, and today I like to share that fact with you. So I firmly believe that the students and scientists and professors will receive some valuable information from my today's presentation. So in the introduction, the carotenoids are found universally in the flowers, in the leaf, in the fruits, and even in the autumn leaf. And because of their enormous functions in plants and animals, currently it is marketed worth $1.5 billion US. So before starting on the carotenoids, let's see what are the plant pigments found in the plant. So The plant pigment can be classified in the four major categories: the chlorophylls, anthocyanins, betalains, and carotenoids. Chlorophylls, as we all know, that responsible for the green color of the plant and which is responsible for the oxygenic photosynthesis of the plant. The anthocyanins are another class of plant pigments. They are derivatives of the flavonoid, water-soluble flavonoid, and they give the deep red, blue, or purple color to the many flowers, leaves, and fruits such as pomegranate, red cabbage, red lettuce, and many more. The betalains are another class of the plant pigment found specifically in the order Caryophyllus plants, such as in cactus, carnations, amaranthus, and beets. So these are the three major pigments we have seen, and now comes to our main topic, carotenoids. So carotenoids are found universally. So when you see the leaf, the carotenoids are hidden by the presence of the chlorophyll. But in the autumn, when the chlorophyll is degraded, the hidden carotenoid pigments are revealed, and that give the beautiful color to our Mother Nature. What is the basic structure of the carotenoid? The carotenoid are composed of simple isoprene units. So in case of lycopene, when 8 isoprene units combine together, it forms the lycopene. And when these terminal side groups are cyclized with the help of cyclase, it may convert to beta carotene, and when it may hydroxylize with the help of beta carotene hydroxylase enzyme, it may turn to zeaxanthin. So in the carotenoid, different functional groups may be attached. It may be like hydroxyl groups, it may be ketone groups, or hydroxyl and ketone groups. So with that much, a significant diversity is existing in the plant carotenoid, and till date, nearly 1,100 carotenoids has been identified from the plants and bacteria. In simplest classification, the carotenoids can be classified in two major categories. The simple hydrocarbon carotenoids, such as like lycopene, beta carotene, or alpha carotene, are classified as carotene. When any functional group is attached to this carotene, they're termed as the xanthophylls. So in xanthophyll, these functional groups may be present in the form of the hydroxyl groups, such as in lutein, the ketone groups like in canthaxanthin, alcohol and ketone groups like in astaxanthin, or the esters of alcohol like in picoxanthin. And they are the carotenoid metabolized product found in the plant like dixin, and they are termed as apocarotenoids. So what is the source of the carotenoids in our diet? When we consume the peppers like yellow peppers or red pepper, they give the beta carotene. When we consume the carrot, they provide us the alpha carotene and beta carotene. The leafy vegetables are the rich source of zeaxanthin, lutein, and beta carotene. The tomatoes are rich source of lycopene, whereas the citrus fruits like orange are the rich source of beta cryptoxanthin. And among the plants, the jackfruit is considered the richest source of carotenoids, which contain nearly 10 times higher lycopene than tomato and 4 to 5 times higher beta carotene than carrot, which is widely cultivated in China and Vietnam and consumed as a traditional food and a traditional medicine and food. Let's have the brief view about the biosynthesis of carotenoid, how they are biosynthesized. So the precursors for carotenoid biosynthesis are formed in MAP pathway or malonate pathway, and these form the first C40 carotenoids, 40 carbon carotenoid, 15 cis phytoin, or simply called as a phytoin. But this 15 cis phytoin is not absorb the light in the visible range. It absorbs the light in UV range. So the first carotenoid which absorbs the light in the visible range is lycopene, and lycopene give rise to the most of the other carotenoids in plants and in bacteria. So in lycopene, from the lycopene, when terminal side groups are cyclized with the help of epsilon cyclase, it gives rise to the alpha carotene. When the beta cyclase work on the lycopene, it forms the beta carotene. and this beta carotene may convert to the beta cryptoxanthin and zeaxanthin. So when one hydroxyl group is attached, it's called beta cryptoxanthin. When two hydroxyl groups is attached on the beta carotene, it's called zeaxanthin. Similarly, the alpha carotene will give rise to the alpha cryptoxanthin and lutein. How we extract the plant carotenoids? So the extraction, the most critical thing is the selection of solvent. So how we select the solvent for the effective extraction of plant carotenoids? So when we have the polar carotenoids in our sample, like violaxanthin, neoxanthin, and lutein or zeaxanthin, when we have this kind of carotenoids, or this called xanthophylls, they are the polar carotenoids, and these polar carotenoids are best extracted by the polar solvent like acetone. And if our sample contains the non-polar carotenes like alpha carotene, beta carotene, or lycopene, they are best extracted by non-polar solvent like hexane. In the special case, the many fruits and the flowers contains the carotenoid esters. What are the carotenoid esters are? The fatty acid is attached with the carotenoid molecule with the help of ester bond. So to one molecule of carotenoid, one fatty acid may attach or two fatty acid may attach. So because of the attachment of this fatty acid to the carotenoid molecule, they behave as a non-polar carotenoid, and they are best extracted by hexane or N-heptane or any other non-polar solvent. So if we want a free carotenoid from this ester, we need to saponify that sample. Saponify means alkaline saponification, like with the KOH. In that saponification, the fatty acid is removed and we get the free carotenoid like lutein. So you may be thinking that if we don't have the prior information about the sample, then how we select the solvent? In this case, we should select the combination of polar and non-polar solvent like acetone, hexane, and ethanol in 1:21 ratio is widely utilized for the extraction of the carotenoid from the sample containing polar and non-polar carotenoids. So when we extract the carotenoid using solvent, we need to add a small amount of synthetic antioxidants like BHT, called butylated hydroxytoluene, and like 0.1% we need to add. It helps in prevent the degradation of carotenoid during extraction. Similarly, when we isolate the carotenoid from our plant sample, it contains the organic acid. So to absorb that organic acid, we add a pinch of magnesium carbonate to minimize the degradation of our carotenoid. And the care should be taken that we isolate the carotenoid in the dim light with the absence of acidic reagents. Now let's move to the functions. What are the functions of carotenoid in plants and animals? So we all know that in the plant, carotenoids are very important for the photosynthesis. And in the photosynthesis, a lots of amount of reactive oxygen species called ROS is generated. So plant carotenoid or this carotenoid have to detoxify that ROS. Similarly, the carotenoids are the source for the production of abscisic acid in the plant. Similarly, it gives rise to the strigolactone, which play a very vital role in the plant microbe interaction. Similarly, due to intense red-yellow color of the flower, it attracts the pollinator and which help in the pollination. In the animals, it plays a very important role as a vitamin A. So many carotenoids are termed pro-vitamin A carotenoid because that convert to the vitamin A in our body. So, and this vitamin is very, very important for animals, and which play the very important role in vision, gene transcription, immunomodulation, embryonic development, bone metabolism, and the skin health. So when you see the like flamingos and the shrimp, there you see the red color. So this red color is not, they are not synthesizing this pigment. These animals are not synthesizing this carotenoid. When the shrimp feed the carotenoid-rich algae, that color accumulate in the shrimp. And when the flamingos eat to the shrimp, this color accumulate in the flamingos. So next time when you spot the flamingos, you should think that your body will be like what you eat. let's have an example of how vitamin A is important in the vision. So in the opsin protein, the retinal is bound in the form of 11-cis retinal. So this bound to the opsin protein and it forms the rhodopsin in the rods of our retina. And when it comes in the contact with the light, it forms the transretinal. And due to this isomerization, the protein conformation change, and that induce the photosensitivity. So the different carotenoids have the different pro-vitamin activity. We'll see in the later slide why they are different. So the all trans beta carotene, or simply called the beta carotene, is considered 100% pro-vitamin A activity, whereas in like alpha carotene or beta cryptoxanthin is considered nearly 50% pro-vitamin A activity. So you were thinking that why different carotenoids has different pro-vitamin A activity. So this pro-vitamin activity depend due to is due to their chemical structure. So chemically, any carotenoid which contain the beta ion ring, I should repeat, any carotenoid which contain unmodified beta ion ring. Unmodified means if any hydroxyl groups is attached on this beta ion ring, it cannot be converted to the vitamin A. So any carotenoid which contain unmodified beta ion ring structure, it may convert to vitamin A, like retinol. It's also called retinaldehyde. So because of the presence of two beta ring in the beta carotene, one beta ring and second beta ring, one molecule of beta carotene can give rise to the two molecules of retinol. So that's it called, it has the 100% pro-vitamin A activity. Whereas in the case of alpha carotene, it contains one epsilon ring, and it contains one beta ion ring. So only this part can be converted to vitamin A. That's why the one molecule of the alpha carotene give rise to the one molecule of retinol, and that's why we consider 50% pro-vitamin A activity. It's so interesting. Now let's see this slide. So we have one molecule like beta cryptoxanthin. As I said earlier, only one beta ring is hydroxylized. So when this one ring is hydroxylized, it cannot be converted to the vitamin A. So only one beta ring have in the beta cryptoxanthin to convert in the vitamin A. So it considered the 50% pro-vitamin A activity. Whereas in case of zeaxanthin, although it has the two beta ion rings, but these beta ion rings are hydroxylized, so it cannot be converted to provitamin A. So how much vitamin A or carotenoid we should eat or demand to meet the demand? That's called recommended dietary allowance. So, and that is calculated the retinol in the retinol equivalent. And the children's like 1 to 3 years of age need 300 retinol equivalent. The man need 900, woman need 700, and when in case of the pregnancy and lactation, this demand increase. And retinol equivalent is considered on the basis of their bioavailability. And one RAE is equal to 1 microgram of retinol, 2 microgram of beta carotene dissolved in oil because it has more bioavailability than 12 microgram of dietary beta carotene or 24 microgram of other provitamin A carotenoid. Why it's higher? We need high because it has lower conversion rate. So just think here that 1 RAE is equal to 12 microgram of beta carotene. So to meet this demand for the man, we need every day to take 10 milligram of beta carotene, and that if we eat the 100 gram fresh carrot or 200 gram spinach or lettuce, that demand can be completed. So you may be thinking that if like zeaxanthin is not the source of provitamin A carotenoids, why they have the importance for the animals? So not only zeaxanthin, like zeaxanthin, ficoxanthin, astaxanthin, and canthaxanthin, the ring is modified. So they cannot be converted to the they cannot be converted to the vitamin A, but they have a great significance for the animal. So what is the significance is? Let's see. So each and most of the carotenoids have the potent antioxidant activity. And as the previous speaker said, the ROS level, very toxic for the cells. So this what carotenoids do, they have to reduce the ROS level in our cells. So due to the cellular metabolism, lots of lipid radicals are formed, and carotenoids help to detoxify that lipid radicals and convert to them non-radical products. So due to that activity, due to the ROS inhibiting activity is protect our cellular lipids, protein, and prevent the DNA from damage. So with that activity, the carotenoids, with that the ROS reducing activity, carotenoids play the very important role in reducing the incidence of cancer, cardiovascular disease, and neurodegenerative disease. Because of the short time, we are not covering this topic, but if you want the information about how the carotenoids are important for this oxidative stress related diseases, then please go through this paper which we published recently. So because of the so many important functions of the carotenoids in the animals, carotenoid has a great demand globally, and currently they are marketed worth $1.5 billion US, and which is dominated by capsaicin, astaxanthin, beta carotene, and lutein. But nowadays, the share of most of this carotenoid is produced synthetically because of the low production cost, whereas only natural carotenoids are share only 24%. And what are the market players for the carotenoids production and marketing? These are like Lycorade, OmniActive, DSM, BSF, and Cryosynthesis are the major market players who are producing and marketing the carotenoids. And carotenoids are incorporated in the eye health products, the poultry feed, aquaculture feed. This poultry feed and aquaculture feed together share the 40% of total market value of the carotenoid. The other are dietary supplement, skin health, and food coloring. In the later slide, we will see why the carotenoids are important in the poultry feed and aquaculture feed. So in the case of the food colorant, we know one of the most famous example, the saffron, widely cultivated and consumed in India as a natural food colorant. And this food colorant is one of the apocarotenoid, quercetin. Now, you may be wondering that why carotenoid is important to the poultry feed. Why so much carotenoids are needed to be added in poultry feed or in aquaculture feed? So in the poultry feed, like in case of the egg, the color, egg yolk color is a key marketing thing which gives information about the health, the presence of health beneficial carotenoid in the egg. So as a symbol of the good marketing, the carotenoid is needed to be added in the chicken feed to get the good yolk color. Similarly, the carotenoids are added in the aquaculture feed to get the intense red color to the aquaculture animals like fish, lobster, or shrimp. So when we grow this fish or the shrimp or lobster in the natural environment, they feed to the carotenoid-rich microalgae. But when we grow this in the captive environment, then we need to feed them the carotenoid. Otherwise, the color will disappear, and that's not be good for their marketing. Let's have an example of the carotenoid-based eye health supplement. So this is a bottle of lutein, which contains the total of just 1.2 gram of lutein, and this 1.2 gram of lutein can be extracted just from 50 to 100 gram dry petals of marigold, which we produce so much in India. So just think that the 50 to 100 gram of dry petals are worth 600 rupees because this bottle is marketed with the price 600 rupees. So I'm saying that you should think that 50 to 100 gram dry petals are worth 600 rupees. Now let's have the brief look about the current research we are doing on the plant carotenoids. So the carotenoid contents are varied significantly among cultivars. So what we do, we screen the different cultivars. Like here we did, they screen the different cultivars of lettuce, and we identified that the red lettuce contained the highest amount of carotenoid, and we suggested that this cultivar should be used to formulate the carotenoid-rich diet. In another investigation, we isolated and purified the major carotenoids from the lettuce, like lactoxanthin, viloxanthin, lutein, and neoxanthin, and we checked their cytotoxicity against the HeLa cell. HeLa, cervical cancer carcinoma cell. So we checked the cytotoxicity of this carotenoid, and we found that neoxanthin is most potent to kill these cells with the IC50 value of just 3.8 micromolar, which is fascinating. In another investigation, we are trying to minimize the use of toxic solvent. Like in case of marigold petals, we investigated to use the cyclohexane or N-haptane, which are less toxic to the human health and even in our nature. So we found that N-haptane can be successfully used to effectively isolate the lutein esters from marigold, and at the same time it may give the higher yield than hexane, which is conventionally used for the extraction of the marigold lutein. So in this work, we isolated the esters, then we purified the lutein from the marigold petal and we characterized with the mass spectrometry. We purified nearly 92% lutein in this investigation, and we checked the cytotoxicity against HeLa cells. And we found that it effectively cytotoxic to the HeLa cell with the IC50 value of the 7.9 micromolar. In the further investigation, we found that lutein from marigold trigger the upregulation of pro-apoptotic protein. Apoptotic means the programmed cell death. As the apoptosis, the previous speaker was telling about the apoptosis. So it's a programmed cell death, the killing of the cancer cell. So it upregulates the pro-apoptotic protein like BAX, P53, and Cas12, and downregulate the anti-apoptotic protein like BCL2 in the cancer cell. So I'm talking about the cancer cell, not in normal cells. In normal cells, the story will be different. In the normal cell, they inhibit the apoptosis. So in the further investigation, we validated with the conformal observation, validated the caspase mediate enhanced expression of this pro-apoptotic factors, and we found the reduce in the mitochondrial membrane potential. So we found enhanced activity of caspase, which is responsible for the apoptosis of cancer cell. And when we use the mito-red and TAPI, the nuclear staining, we found the lower in the activity. In the further investigation, we found that lutein from marigold trigger the DNA damage of HeLa cell, responsible for the apoptosis. So when we treated the lutein with a concentration of 10 micromolar to the HeLa cell, we found more than 90% cells are tunnel positive. So with this investigation and with the information from the prior studies, we developed this hypothesis that lutein induce the pro-oxidant actions in the cancer cell. Pro-oxidant action means against the antioxidant. So the previous speaker was telling these are the bioactive compounds are antioxidants. So carotenoids are also a bioactive compound which play the antioxidant role in the normal cell. But in cancer cell, they play pro-oxidant role. And due to this role, it activates the tumor suppressor protein, which suppresses the tumor, like P53. And this P53 upregulates the expression of pro-apoptotic protein. And this pro-apoptotic protein forms the pores in the outer mitochondrial membrane, responsible for the increased permeabilization of the mitochondria, and which cause the release of cytochrome C and caspase-mediated apoptosis of HeLa cell. So the apoptosis means killing of cancer cell effectively. In another investigation, the beta-cryptoxanthin isolated from the citrus also showed the similar apoptotic activities against HeLa cancer cell. In this study also, we found the caspase, even the cleaved caspase, which is called the active caspase expression, is upregulated in the cells treated with beta-cryptoxanthin, responsible for the release of cytochrome C and caspase-mediated apoptosis. So these are our studies I discussed you with briefly, and now we'll have what are the current trends in plant carotenoid research. So we see the few trends. The first trend is agro-industrial waste utilization. For example, the lycopene recovery from tomato processing waste. So in the tomato processing waste, like first we see what is tomato processing. Tomato is used to produce the puree, paste, pulp, serum, and syrup, and it generates a lots of amount of tomato pomace, which contains seeds and skin rich in lycopene, and this can be utilized in the lycopene recovery. Similarly, in the current time, the citrus processing waste are utilized for the isolation of economically vital carotenoid beta-cryptoxanthin. Another trend is the genetic engineering of carotenoid biosynthetic pathways in the staple food crop. We know the most famous example of the golden rice, produced almost 16 years back, and now we have the good news that the Canada, United States, Philippines, and Bangladesh, and many more countries has approved the golden rice for the cultivation in the field. So soon we'll see this golden rice in our field too. And after the 16 years of production of the golden rice, we have produced many more golden crops like cassava, maize, wheat, canola, banana, carrot, orange, potato, soybean, pepper, lettuce, and tomato we have produced. The current trend is exploring the underutilized source of carotenoid. We have seen the jackfruit, very highly rich in the lycopene and beta-carotene, but not widely utilized globally. Another example in case of India is the Tinospora cordifolia. It's called giloy, very famous nowadays because of the COVID medication which suggested the enhanced immunity. So the berries of the Tinospora cordifolia contain almost 10 times higher lycopene than tomato, which is considered one of the richest sources of lycopene. So we are working on to explore this underutilized source, which can be used to formulate the carotenoid-rich diet. Another trend is microencapsulation, which we are currently working on that. Why we do the microencapsulation? Because the carotenoids are very much sensitive to the light, the temperature, pH, metal, and oxygen-mediated degradation. So what we can do is we can encapsulate these carotenoids inside the beta-cyclodextrin or any other encapsulating agent, and it can improve the storage stability. So these are the current trends that we discussed, and now let's see what are the future perspectives in the plant carotenoid research. So we have seen in 2018 and 19, the carotenoid market was worth $1.5 billion, and it has been expected to reach $2 billion in 2026, and the demand for natural carotenoids are increasing because of the health effect of the synthetic carotenoid. Synthetic carotenoid may be having some health issues, so people are considering the natural carotenoid for their diet. So the demand of natural carotenoids are increasing, and this demand will further increase in the future. What is another future perspective in the carotenoid research is the solvent-free extraction. Generally, the carotenoids are extracted using the solvent, and that may have the residual effect in the final product. What we can do, we can extract the carotenoid using CO2 in the supercritical stage, and the CO2 can be recovered again, and then we have the carotenoid without any residual solvent. So the current time also, lots of people are considering this technology, and we expect that in the future, this technology will further improve. Another perspectives, in case, especially for the India, what we are suggesting is the recycling of India's floral waste. In India, we produce 1,000 tons of floral waste every day, and that is the problem of the disposal and environmental sustainability. So what we can do is we can isolate the economically vital carotenoid from the floral waste. That's mostly like marigold. So we can use to isolate the lutein from that one, which may have their recycling and environmental sustainability. At the same time, it may generate the significant amount of income. The another future perspective is the screening of or crop genetic resources. We have large amount of cultivars which we have not screened so far. So we screen that cultivar and we identify the carotenoid-rich cultivar, so which can be used to isolate the carotenoid to formulate the diet rich in the carotenoid, which has the greatest scope. Another future perspective is the understanding the molecular mechanism of the protective effects of carotenoid in cancer and other chronic diseases. Other chronic diseases means the cardiovascular diseases, neurodegenerative diseases. There are so many chronic diseases carotenoids are used, but the mechanism we don't know. For example, the carotenoids are responsible for the apoptosis of cancer cell, they reduce the multi-drug resistance, it inhibits the cell cycle, means it arrests the cell cycle, it inhibits the metastasis. Metastasis means the migration of the tumors. So it inhibits the metastasis, and carotenoids are effective inhibitors of NF-kappa beta factors, which are considered NF-kappa beta factors or FOS or junk, and they are considered the tumor promoting agents. These tumor promoting agents are effectively inhibited by the carotenoid. Thus, it inhibits the proliferation of cancer cells. So we know this effect is triggered by the carotenoid, but the precise mechanism is still not known. So the future perspective is to precisely identify the mechanism how the carotenoids triggering that effect. For example, we have seen that lutein trigger the pro-oxidant effect, and it induce the ROS in the cancer cell, and which activate the p53, responsible for the apoptosis of cancer cell. But we still don't know how the p53 is actually activated by the carotenoids. So such type of mechanism or in the molecular levels are needed to be elucidated in detail. So with this my talk, I like to summarize that plants are really a hidden treasure of economically important and health beneficial carotenoid. The global demand for natural carotenoids is increasing, leading to the ample opportunities in production and marketing. The current research is focused on enhanced production of the carotenoids with utilizing minimum resources, industrial waste utilization, and their eco-friendly extraction. So the plentiful utilization of carotenoids in the diet should be promoted to minimize the oxidative stress in the body, leading to the protection from chronic diseases. Thank you so much for listening.
Thank you very much, sir, for your enlightened talk. Now we'll take up a few questions. The first question is from Anandita Banerjee. In lower group of plants, what are the major roles of carotenoids?
In the lower plants, or even in the microalgae or the seaweeds, the primary role in the plant is the photosynthesis, and it helps to capture the light, and it transfers the chlorophyll for the photosynthesis. At the same time, because in the photosynthesis, the lots of amount of the ROS is formed, and it helps to detoxify that ROS. So that's the major function in the plant or animal or in the microalgae.
Okay, sir. Another question. What is the simplest and best procedure for estimation of carotenoid in 1 gram of flower petal?
Okay. So if the simplest method is the photometric method, if we have the carotenoid, like the spectrophotometric method can give the information about the total carotenoids in that sample. So how much carotenoid is there in that petal, but we don't know what are the carotenoids in that petal. So if we want to know just how much carotenoids total, like in the total of lutein and zeaxanthin or beta carotene, that total can be estimated by the spectrophotometric method, where if you want their composition, like how much lutein and how much zeaxanthin or how much beta carotene, then HPLC is the most widely used technique for that.
Okay. Question is from Sukanya Chakravarti. What are the pigments present in hydrangea?
Sorry, I don't know what is that plant is, hydrangea. So when you see any plant or any microalgae, like lutein, neoxanthin, veloxanthin, lutein, chlorophyll A, chlorophyll B, or chlorophyll C in case of the the seaweed. So like neoxanthin, veloxanthin, lutein, chlorophyll A and chlorophyll B and beta carotene are universally present in all green leaf. So if I know that information about that sample, I can tell you the possible composition of the carotenoid in the plant. So this is my email ID, or this is the research scholar ID. You please mail the details of that plant, and maybe I can suggest the extraction, how we can extract that sample, and what may be the possible composition of the carotenoid in that sample.
Okay, thank you, sir. Another question. What is the role of carotenoids in stress?
It is stress of I should know the stress in the plant or in animals. I should go with like animal stress. As the previous speaker said, that we all have the oxidative stress. We all, maybe most of the cells, if I have the oxidative stress, the carotenoids have to reduce that oxidative stress. How it reduce? It plays a very important role in detoxifying the free radicals. So the carotenoids are the potent antioxidant molecule, and in the cell, it bound with the cell membrane. When the cell membrane, the lipid radicals are formed, and these carotenoids have to detoxify the lipid radicals. So ultimately, it reduces the RS level and oxidative stress in the cell. And similarly, if you go in the plant, the carotenoids are the source of abscisic acid. So that abscisic acid play a very important role in the stress, like in water stress.
Okay, sir. Another question. Is there any carotenoid crossing blood-brain barrier so as to treat neurodegenerative disease owing to their tremendous antioxidant potentialities?
Yeah, that's very good question. There are some carotenoids which can cross the brain barrier. Like we are testing some carotenoids against the brain cancer cell, like UMG 251, and we found the significant cytotoxicity of that cell. When we found the significant cytotoxicity against that cell, means we can think that our carotenoid is crossing that barrier. And we hope that we'll get very good results with that experiment, so which we are currently doing on that.
Okay. Kirtana MS has asked, is it possible to extract carotenoid pigments from algae?
Yeah, algae is one of the richest source of carotenoid, and industrially feasible. We can cultivate, and that procedure is really industrially feasible, and lots of demand is there. A lot of research is there. I did my PhD from CFTRI Mysore, and in our department, the cultivation of the department name is the Plant Cell Biotechnology, and their major work is to produce the algae for the biofuel, for the carotenoid, for the astaxanthin, and many more carotenoids. I want to give an example of like Botryococcus or and there are many examples which contain a significantly high amount of beta carotene and astaxanthin and other carotenoids. Sorry, now I remember the Dula is the algae contain so much carotenoid. I think higher among all the algae.
Will carotenoid be spoiled if we cook carotenoids contain veggies like carrots?
Yeah, that's a very good question. So we have the carrot, and when we have the carrot, spinach, and lettuce, we cook that one, and during the cooking procedure, during heating procedure, some carotenoids will degrade, but at the same time, their bioavailability will increase. So when you eat the raw carrot, it contains more carotenoid, but they are less bioavailable. Bioavailability means when we take the carotenoid, how much carotenoid it absorbs in our body. So that's called bioavailability. How much carotenoid is bioavailable? So when you eat the raw carrot, it has more carotenoid but less bioavailability. When you eat the boiled carrot, it has a little less amount of carotenoid but more bioavailability. So there's no problem if you cook and boil them, but we should not throw that water and because it contains so much of other bioactives. So it's not a bad idea to cook the vegetables. It's a good idea.
Okay, thank you, sir. Another question. Can consumption of excess vitamins, carotenoids, or multivitamin tablets cause digestion disorders, PCOS, etc.?
So for the healthy human being, the higher consumption of the beta carotenoid doesn't create the much problem for the healthy animals. I'm not going to about the vitamin A, I'm telling about the carotenoid. So when you consume the more carotenoids, your skin color may change. So only that is a physiological problem, but it doesn't create much problem at the cellular level. So, but in case of like the person who is smoking should not consume the beta carotene supplement. So because they are smoking, and if they take the beta carotene supplement, they may have the higher incidence of the lung cancer. So we should think about that.
Okay, next is from Sandhya Panikar. Why carotenoids need to be consumed with a fat?
Yeah, that's a very good question. The first thing that the carotenoid is the water-soluble molecule. It's not a water-soluble. We know that some vitamins are the water-soluble, like folic acid, the phenolic compound are water-soluble, and carotenoid and the tocopherols, like other vitamins, are the fat-soluble. So the bioavailability of the carotenoid is increased when you take with the fat. So when you eat the carotenoid, it mix with the bile salt and form the micelles, and that micelle have to absorb that carotenoid in our body. So when you take the carotenoid with the more fat, the bioavailability is certainly increased.
Okay, what is the anti-carcinogenic potential of carotenoids?
Means anti-cancer potential. Yeah, it has a great, as I said, carotenoid has a great anti-cancer potential, and a lot of research is going on that one. But so far, the clinical evidence doesn't support much, so we need the more research on that one. We need the more clinical research, and hopefully in the future we have some drug, the potent anti-cancer drug based on the carotenoids.
Okay, another question from Renu Shri. Can we extract carotenoids from fungi?
Yeah, there are many fungi contain the carotenoid, and especially the genetically engineered fungi are used for the commercial production of the carotenoid. Please go through my research, and you'll find some papers which we work the carotenoids from the fungi. So there are also research of fungi, and we can extract the carotenoid from that one like plants.
Okay, another question is, do carotenoid exert any other pathway other than apoptosis for killing cancer cells?
Yeah, as I said in one of my slides, that the carotenoids can inhibit the phosphorylation of many proteins. So that's phosphorylation like POS, JNK, NF kappa beta. It can inhibit the cancer-causing factor, and similarly, it can reduce the pro-inflammatory mediators like interleukins. So cyclooxygenase. So it can inhibit the interleukins. It can reduce the oxidative stress in the body, and ultimately it can reduce the chances of incidence of cancer in the body. So the first thing is the antioxidant thing. The carotenoids are the potent antioxidant, and due to potent antioxidant activity, it reduces the oxidative stress, and due to reduction in the oxidative stress, it helps to minimize the incidence of cancer. At the same time, it reduces the the pro-inflammatory mediators like interleukins in the body, and that helps to the that helps in the reduced incidence of cardiovascular diseases.
Okay, another question from Arkaja. Sir, for genetically modified plants, can the chemical nutrition provided by carotenoids be affected?
Can you please repeat that question?
Yes. Genetically modified plants, can the chemical nutrition provided by carotenoids be affected?
So actually, this question I'm not understanding, but I want to I want to give the appropriate answer. What I can say is in case of the golden rice, we have enhanced the beta carotene in the rice. The beta carotene is there's no presence of the beta carotene or presence in very little amount. So we enhance certain genes to proliferate the synthesis of beta carotene in the golden rice, and we enhance 50 times or 100 times. But because we are targeting only some particular gene, they are not affecting the other metabolites, because the rice is the resource like oryzanol, flavonoid, the starch, like primary metabolite. So these primary metabolites and secondary metabolites are not affected by this genetic engineering approach. So when we modify any other plant, we target the particular genes, and that particular genes affect the particular metabolites and their precursors. So I think if you get the answer that the genetically engineering any crop plant doesn't affect the other metabolites.
Okay, last question, sir, from Pooja Baruha. Sir, since carotenoids does not come out well in presence of chlorophyll, still some plants imparts in the presence of chlorophyll. How?
Sorry, I didn't get that question.
I'll repeat, sir. Since carotenoids does not come out well in presence of chlorophyll, still some plants imparts in the presence of chlorophyll. How? I think she means carotenoids, how it will be present along with chlorophyll, what I understood, sir.
Yeah. That question also actually needed to explore again. So each and every leaf contains some carotenoid, like I said earlier, like neoxanthin, villoxanthin, lutein, and beta carotene with the presence of chlorophyll A and chlorophyll B are universally found. So these carotenoids are hidden by the chlorophyll, but in some of the flowers, like in marigold petal, due to the absence of the chlorophyll, we can see the carotenoid. Similarly, in the carrot, there's no presence of the chlorophyll, we can see the carotenoid. So if you see any green leaf, and this appearing green means is not the mean that doesn't contain the carotenoid. It contains the carotenoid, but they are hidden by the chlorophyll. And in the autumn, the chlorophylls are degraded in the autumn, and then the hidden carotenoids are revealed.
Okay, I think we have covered almost all the questions, sir. Thank you very much, sir. On behalf of Mount Carmel College Management and Department of Botany, Mount Carmel College, I extend a warm thank both the speakers, Dr. P. Vishweshwara Rao and Dr. Ramesh Kumar Saini, for your valuable information and excellent talk. Thank you very much, sir. Over to Miss Jayanti.
Thank you, Savita ma'am. Dear participants, this is regarding the feedback form. The feedback link is shared on the YouTube WhatsApp chat. It was shared at 3:40, that is, we have given almost 20 minutes for all of you to access the feedback form and submit. We will also wait for another 10 minutes in case if anybody has not submitted, after which the link will not be available. You cannot access the link anymore after 4:10. So kindly make sure you submit the feedback form for today so that you can put your attendance for day 2. Thank you very much. Savita ma'am, you can take over, ma'am, to end the session.
Yeah, thank you very much, sir, for your excellent and informative talk.
Thank you so much, ma'am. I should say great thanks to the organizer for considering me. And any students have, if any question is there, please mail to me or send a message to my ResearchGate profile or any other medium. I will try to help them. at my best.
Yeah, thank you very
Thank
much,
you
sir.
so much. Yeah.
Yeah, thank you very much.
So thank you very much. Today's session we will end then. Thank you, sir. Thank you, teachers. Thank you all the speakers who have joined us today.
Yeah, Jayanti, can we leave now?
I'm ending the session. 1 minute, ma'am.
Yes, yes.
Thank you.
Yeah.